The cell cycle is a series of events that are carried out in a predetermined sequence in order to duplicate a cell that already exists. Imagine what will happen if these events are carried out in a random manner. The answer is the consequence will be disastrous. So, in order to make the entire process of cell cycle highly accurate and reliable, cells possess a specific regulatory system known as cell cycle control system. Hello and welcome to SAM Biology class. Friends, today we shall discuss about cell cycle control system including checkpoints, cyclins and cyclin dependent kinase. The cell cycle control system operates much like a timer and trigger events of the cell cycle in a set sequence. This control system is based on a connected series of biochemical switches, each of which initiate a specific cell cycle event. The system of switches possesses many engineering features which increase the accuracy and reliability of cell cycle progression. First, the switches are binary. They are like on or off switches that trigger cell cycle events in a complete irreversible manner. Imagine what will happen if events like chromosome duplication and nuclear envelope breakdown are partially initiated or started but not completed. It will be a blunder to do so. Second, the control system can operate effectively under a variety of conditions. Third, the cell cycle control system is highly adaptable. It can be modified to suit to specific cell types or to respond to specific extracellular and intracellular signals. In most of the eukaryotic cells, this control system triggers progression of the cell cycle at three major checkpoints, which are: number one, start or restriction point. This is the first checkpoint. And second one is G2M checkpoint. And third one is metaphase to anaphase transition. This figure shows the different checkpoints that are part of the cell cycle. One is start checkpoint, which is also known as restriction point, where the cell commit to cell cycle entry and chromosome duplication. Second checkpoint is G2N checkpoint, where the control system trigger pre-mitotic events leading to alignment of chromosomes on spindle in metaphase. And the third checkpoint is metaphase to anaphase transition, where the control system stimulates sister chromatid separation leading to the completion of mitosis and cytokinesis. The cell cycle control system verifies whether the condition is favorable for cell cycle progression. It also verifies whether the preparation is complete to do so. The control system blocks the progression of cell cycle at any of these three checkpoints if it detects any problems inside or outside the cell. The central components of the cell cycle control system are members of a family of protein kinase known as cyclin dependent kinase or CDK. The activities of these enzymes rise and fall as the cell cycle progresses. This leads to a cyclical change in the phosphorylation of specific intracellular proteins which initiate or regulate major events of the cell cycle. The cyclical change in the activity of cyclin-dependent kinase are controlled by a complex array of enzymes and proteins. The most important of the CDK regulators are a group of proteins known as cyclins. As their name indicates, cyclin-dependent kinases are dependent on cyclin for their activity. Specific cyclin bind with cyclin-dependent kinase to form cyclin CDK complex, which is partly active. The cyclin CDK complex then undergo further changes to get fully activated, and the fully activated form then phosphorylate various intracellular proteins, which in turn initiate or regulate major events of the cell cycle. Cyclins are a group of proteins which undergo a cycle of synthesis and degradation in each cycle, whereas the level of cyclin dependent kinase remain constant. The cycle of synthesis and degradation of cyclins result in a cyclical change in the assembly and activation of cyclin CDK complex, which then trigger cell cycle events. There are four different classes of cyclins. All eukaryotic cells require three of these. First one is G1S cyclins that activate CDKs in late G1. It helps trigger progression through the start that result in commitment to cell cycle entry. Its level fall in the S phase. Second one is S cyclins that activate CDKs soon after progression through start and help stimulate chromosome duplication. The level of S cyclins remain elevated until mitosis. Third one is M cyclins that activate CDKs that stimulate entry into mitosis at G2M checkpoint. In addition to this, in most of the cells, there is a fourth class of cyclins known as G1 cyclins that help govern the activity of G1S cyclins that help trigger progression through the start. Friends, you know that start is the first checkpoint in the cell cycle. Activation of cyclin dependent kinase. In yeast, there is a single cyclin dependent kinase which binds with all classes of cyclins and trigger different cell cycle events by changing cyclin partners at different stages of the cell cycle. But in vertebrates, there are four different cyclin dependent kinases, of which two interact with G1 cyclin, one with G1S and S cyclin, and the remaining one with M cyclin. Study of the three-dimensional structure of cyclin and cyclin dependent kinase reveals that in the absence of cyclin, the active site of CDK is partly blocked by a slab of protein. Binding of cyclin with the cyclin dependent kinase result in the removal of the slab of protein away from its active site, thereby partially activating the cyclin dependent kinase. After this, full activation of cyclin dependent kinase occur when another enzyme known as CDK activating kinase phosphorylate an amino acid near the entrance of the active site of CDK, thereby further increasing its activity. Phosphorylation by CDK activating kinase result in a small conformational change in the cyclin dependent kinase, thereby further activating it, after which the activated form of CDK cyclin complex phosphorylate specific target proteins which then trigger different cell cycle events. The rise and fall of cyclin levels is the primary determinant of CDK activity. However, additional regulatory mechanisms fine-tune the activity of cyclin-dependent kinase. Now we shall discuss about all these additional mechanisms which regulate the activity of cyclin-dependent kinase. First, phosphorylation of a pair of amino acids in the roof of kinase active site inhibit the activity of cyclin-dependent kinase. Phosphorylation of the same amino acids by protein kinase called V1 inhibit the activity of cyclin CDK complex, whereas dephosphorylation of these amino acids by phosphatase called CDC25 increase the activity of cyclin dependent kinase. In addition, there are some specific proteins called as CDK inhibitor protein CKI, which also inhibit the activity of cyclin dependent kinase. Three-dimensional structure of cyclin CDK CKI complex reveal that binding of CKI with CDK stimulate a large rearrangement in the kinase active site, thereby rendering it inactive. Whereas the activation of specific cyclin CDK complex drive the progression of cell cycle through start and G2M checkpoint, the metaphase to anaphase transition is triggered not by protein phosphorylation but by protein destruction, leading into the final stages of cell division. The key regulator of metaphase to anaphase transition is anaphase promoting complex of cyclosome, which is a member of ubiquitin ligase family of enzymes. The anaphase promoting complex catalyzes the ubiquitination and destruction of two major proteins. One is securin, which protect the protein linkage that hold the sister chromatids together. Destruction of securin at the metaphase to anaphase transition activate a protein kinase which separate the sisters and unleash anaphase. The other targets of cyclosome are S and M cyclin. In addition, there is another ubiquitin ligase called as SCF, which ubiquitinates certain CDK inhibitor proteins in late G1 and help controlling the activation of SCDK and DNA replication. Finally, the cell cycle control system also depend on transcriptional regulation. So friends, this is all about cell cycle control system. For more updates, please subscribe my YouTube channel Sam Biology Class. Thanks for watching.